Beyond biochemical pathways, regulation of protein synthesis by prokaryotic genes. Prokaryotes are the masters of metabolism in the living world. The prokaryotic genome typically consists of a single circular chromosome in which protein-coding genes are arranged in tandem along a very long double helical molecule of DNA. DNA replication speed is at a premium among prokaryotes because natural selection favors single-celled species that can reproduce more quickly than others. Prokaryotic selective forces favor 1. Keeping the genome as short as possible, as well as 2. Conserving resources such as energy and raw materials. That way, cells can replicate their DNA and grow as quickly as possible, depending on available resources. Ideally, proteins should be synthesized only when they are needed. For metabolic pathways required to break down sugars, for example, the protein chains that make up the enzymes in those pathways should only be synthesized when they are required. Some bacteria, such as the E. coli that play a symbiotic role in human digestion, have evolved the ability to utilize either glucose or lactose sugars as their sources of energy and carbon skeletons. It follows that natural selection should favor the ability to control the production of lactose versus glucose metabolizing enzymes. Bacteria that can turn enzyme production on and off have a selective advantage. This is accomplished by an innovation known as the Prokaryotic Operon. The concept of the Prokaryotic Operon was first proposed in 1960 by a pioneering team of French scientists. Operons usually consist of a regulatory sequence of DNA known as an operator, followed by a series of protein-coding genes required to make the enzymes of a specific metabolic pathway. When the operator is bound to another protein known as a repressor, this blocks the RNA polymerase from transcribing the genes so that mRNA cannot be made. Operators are controlled by repressors and/or inducers that act as on or off switches for the production of mRNA. In 1965, François Jacob, André-Michel Lwoff, and Jacques Monod won the Nobel Prize in Physiology and Medicine for this groundbreaking work.